And good evening, ladies and gentlemen. It's great to see some familiar faces, but it's equally great to see some new faces in the audience. And obviously, I'm going to try and present in a way that those of you who have not heard anything about AGL can get a reasonable picture of what our business is about. And that is addressing, in fact, on a diagnostic level, the same sort of area that Satu was talking about, the rise of cancer amongst our community, and making sure that we have the right types of treatment to actually make a difference in those cancers. And I should say, by the way, that because I did get asked in one of these presentations, are you a listed company? Yes, like the other companies here, we're listed on the AIM market. We also have an ADR, which is trading on the OTCQX in New York as well, as a precursor to a possible NASDAQ sometime in the future. And what I'm going to be talking about here is the use of a very simple and quite elegant engineering solution to a major medical problem. Oops, sorry, I should have explained more about this. So the thrust of what we're trying to address is the fact that everybody's individual cancer is different. It's very easy to assume that you've got 3 breast cancer patients, they all have the same disease. And I guess that used to be my presumption before I got more involved in this field. Fact of the matter is that cancer is a heterogeneous disease. It's made up of lots of different components, and each patient will have a different disease. And what's more complicated than that is they'll have a different disease within the individual patient during the course of their disease. In other words, the cancer mutates and changes whilst the patient's being treated. So this makes it phenomenally difficult, actually, to provide a good treatment, because if all these patients have got different diseases, how do you know which drug might be suitable for which patient? And as Satu pointed out, quite often these very expensive drugs, which might be costing in the region of £150,000 a year for the patient, only work in about 30% of the patients. So the question is, which is the 1 in 3 patient that this drug's going to work for? And which are the 2 in 3 where not only is it a waste of money, but they're suffering quite nasty side effects from the treatment which is not working for them? And that's what we talk about when we talk about personalised medicine or precision medicine. It's about understanding that the patient's cancer is different and trying to give them a drug which will work for them. And the big pharmaceutical companies are spending predominantly all of their efforts now targeting the individual pathways of different types of the disease in accordance with the different mutational status. So that, for example, is a very well-known, Erbitux is a very well-known drug for colorectal cancer, but it's only applicable for KRAS negative patients, and that's a sort of technical term, but basically you need to know, is a patient KRAS negative or positive? And there's lots of the similar things, the lung cancer, EGFR, breast cancer, HER2 that we already heard about. So how do they do that? Well, the traditional way of doing it is through a solid biopsy, and that works perfectly well. So in the colorectal cancer example, the patient has a colonoscopy, and if they find a lump, they cut it out and they take it away to a laboratory. So they basically physically cut out some of the cancer, and then they've analysed it. And by doing that, they can look at the individual cells and work out the molecular status and make some initial decisions about how best to treat the patient. And that works fine if you can access the tumour. Some tumours, it's very difficult to access them. So, for example, if there's a brain tumour, you're not going to cut into the brain to access the tumour for the purposes of biopsy. Quite often you will do that as a surgical resection to reduce the physical presence of the cancer. So some of these cancers are difficult to access. But the really fundamental point, which is dead simple, is once you cut something out, it no longer exists. So you can't cut it out twice. So somebody has breast cancer, they have the lump cut out, or maybe they have a mastectomy, they've still got cancer, and that cancer is mutating and changing. But now you go back to do a second biopsy, and you can't, because you've already cut the lump out. So what do you do then? Well, the answer is you struggle, and that's why we hear of people who have had what appears to be a successful progression in their treatment. Their cancer size has reduced in size and everybody gets quite happy. But then, unfortunately, they start regressing and the cancer becomes chemo-resistant, meaning that the drug doesn't work anymore. And the doctor really needs to find out what's happened here, because the cancer's changed and the original drugs are not working. Well, but how can they do that? Because the lump's already been cut out. And I'll show you later that one of the things they try to do is to biopsy the metastatic cancer. But obviously by this stage, the patient's cancer has spread and they've got secondaries, and they're now trying to find a solid biopsy from a difficult, inaccessible place to get this clinical information. So what we've done with ANGLE is we're pioneering a new approach, which is known as liquid biopsy. So what we're trying to do is to get cancer cells not by cutting them out of the physical tumour, but actually by a simple blood test. And this works for a very interesting medical reason, which is that the patient original cancer tumour will drop cancer cells in the bloodstream, and they go around inside the patient, and they're called circulating tumour cells. And the purpose of those cells, their existence, is to land somewhere else and cause a secondary cancer, which ultimately is the thing that kills most cancer patients. So there's a small number of cancer cells, very, very small number, but if you could get hold of them from the bloodstream, then you've got a simple blood test. And these patients are having blood tests all the time. That's non-invasive. It can be repeated whenever you want, and it's relatively low cost. And if you can get an intact cancer cell, you can get the same medical information as you might otherwise get from a solid biopsy. So that's the crux of what we're seeking to achieve with our system. And it's based on a very, very simple micro-fluidic cassette, which is this. So that is a one-time use consumable. It's made out of a polycarbonate. It's been designed, obviously, extremely carefully. It's owned by ANGLE, so we have a series of patents covering this. And it's manufactured to plus or minus 1 micron. That's 1/1,000,000th of a metre, so 1,000th of a millimetre in dimension. And this device can capture the cancer cells from the patient blood by flowing the blood inside the cassette. And the reason it can do that, I will show you in this little animation. So that is the inlet port, and the blood flows inside the cassette and comes out of the outlet port. And those loops in there, and I can show you this afterwards in drinks if you'd like. We've got the series of steps here. That's where the main core pattern exists. We've got the red blood cells which just stream over through the gap, and the white blood cells as well. The large cancer cells there you can see are 3 or 4 times larger than the white blood cells, and they don't get through the gap. So it's actually quite a simple idea. It's a filter system based on the size of these cells. And another important characteristic, which is that they're less compressible. So if you imagine trying to squeeze through a gap, if you're less compressible, a bit like a golf ball plays a squash ball. You can push a squash ball through a gap, but you might not be able to push through the golf ball. So that is the cancer cell that's caught there. And this is what it looks like inside a cassette with real blood. So this is 100 times magnification. And those lines there are actually the steps that I was showing you before. And you've got thick red, dense red blood cells and white blood cells streaming across here. That's the critical gap, so there's only 1 cell deep, which is why it's a lighter coloured red. And in a minute, you're going to see the absolute core of our technology, which is the capture of a cancer cell in this device. And that's it there. Now, the great thing about that is it's having absolutely no impact on the flow of the blood. So this system, unlike some competing systems in membranes and various technologies, does not clog up. And you're left with the cancer cells sitting on our critical step. And that's it. It's as simple as that. Now, obviously, to make this kind of system effective, you need an instrument to run it. And that's what we've developed over a period of years, which is a fully automated instrument. And that's, the lighting's not particularly good, but what you should see in there is a tube a bit like that, which is a standard blood tube inserted into our system. So the user, the nurse draws blood in exactly the normal way from a peripheral vein. It's in a standard EDTA tube that they use in the hospitals on a normal basis. Just untwist the top and twist it onto our instrument and hit the button. And our system will do everything else. So it will automatically run the blood through. There's one of the cassettes, and that's sitting in a purpose-made clamp, which makes sure the tubing goes in the right place. So it will run straight through the cassette. Now, I showed you what it looks like where the cells sit on the step. Basically, we can reverse the flow by changing that valve and just wash the cells back out. So we provide cancer cells in a test tube. And that's a really, really important facet of what we're doing. We are basically giving the same output as a solid biopsy, but actually it's pure cancer cells with a few white blood cells sitting in a little bit of liquid, ready to be analysed in the same way that they're used to analysing cancer cells. And that's an important part of our proposition. So I told you this is an emerging market. This isn't something that AGL has made up, though. This is a big market that's coming through. This is going to whether or not AGL's successful is a different question, but I'm absolutely certain that liquid biopsy is going to be the way of the future in treating cancer patients and indeed certain other types of patients. And this is a Goldman Sachs report that's recently come out, and they have estimated the market size to be $14 billion by 2025. That's the US market alone. It's quite interesting. It contrasts with the number that we estimated of £8 billion, pounds rather than dollars, in the global market. So they think it's roughly 50% larger than our previous estimates of the market size. Whichever number you accept, and it could be something completely different, but in the same ballpark, there are four major market segments where this can be used. So screening people to find out whether they have cancer early. Early detection is one of the major things that's needed in cancer treatment. Therapeutic decision making, I mentioned about choosing the right drug for the patient at that time in their disease. Minimal residual disease, so this is, you've been treating a patient, have you actually got rid of the cancer or is it still there? Post-treatment monitoring, one of the most important issues is that a patient who's in remission, there's currently no satisfactory way of determining whether they're having a relapse. And that's highly dangerous. The number of times we hear about friends who've been in remission and suddenly relapse and then they're dead very, very soon afterwards. We're hoping that the liquid biopsy can enable a routine blood test on a periodic basis just to check that patient doesn't need any more treatment. So there's lots of different things there. There's two different ways of doing liquid biopsy. There's something called ctDNA, otherwise known as cell-free DNA or circulating tumour DNA. That's fragments of dead cancer cells in the bloodstream. We don't do work on that. We do work on circulating tumour cells, which are whole, intact, living cells that come out. They're the ones that cause a secondary cancer. And the Goldman report points out that these cells offer a particular advantage because they give access to the full cellular morphology and genetic content, such as RNA. That's a direct quote from their report. However, and this is the key point, they come up with a further comment, which is that CTCs are exceedingly rare and more difficult to isolate than ctDNA. And that has been the situation for many years. It's been almost impossible to get these CTCs because there's only one cancer cell in 1,000 million blood cells. But I've just shown you a very, very simple technology which can change that paradigm. And we are finding in study after study that we can get hold of these circulating tumour cells from pretty much every single patient that's tested. And previously it was more or less impossible to get them from 2/3 of the patients. So we're changing that paradigm, and we believe that we can therefore play an important role in this liquid biopsy market as it emerges. We've got a very strong competitive differentiation against multiple other companies that are addressing this market. And the major attributes here, which have been backed up by 5 independent cancer centres' evaluation of our system, all of them consistent. It's a simple system, it's easy to use, it works well, it's low cost. It works on all cancers. We don't have to check. Most systems, you have to change them depending on what type of cancer you want to get. Breast cancer, lung cancer, colorectal. Our system, no change is needed. And furthermore, it captures all of the different types of cancer cells, including the mesenchymal cells, which cause a secondary cancer. It enables an easy harvest. The big problem that has been with some of the early systems that tried to capture circulating tumour cells was a complete inability to harvest the cells for analysis. So traditional techniques, you could count how many cells there were and find out how quickly the patient's going to live or not. But you couldn't analyse them and work out how to treat them. It's obviously much, much higher value if you can actually improve the patient's treatment. In terms of the path to commercialisation, we've got a well-structured approach to that, and we've moved very strongly through it. Indeed, announcement today with regard to the research sales, which I'm going to come on to in a second, is a big step there. The core of what we're doing now is working on regulatory authorisation. We have a CE mark in place for Europe, so that we've got government approval to use this for treating patients in Europe. We're working on getting a matching approval in the FDA for the United States. That's in process. We've been working very hard on it for well over 18 months now. We hope to be the first company in the world to be officially authorised by the FDA to do this kind of work, particularly for harvesting the cells for analysis. We're working on translational research, which is proving how well the patients can do as a result of this kind of approach. So that's getting people to use our system for the benefit of patients. And that requires us to do work with major cancer centres. It's not an internal thing. You need big cancer centres to do that work and demonstrate benefit to patients. So research use sales, this is an aspect, we made a commitment earlier this year that we would deliver our first sales for research use in this calendar year. We've achieved that. It was announced this morning, and we're very pleased with the progress there. This is a significant market, around £250 million market per annum. And we believe that it's got two parts to it. There's the cancer research centres who are doing basic research, and we've been selling into them. And there's also the pharmaceutical drug trials. So there's a prospect of using this system in trials of new drugs to work out which patients will respond, amongst other things. And that marketplace, we think, is potentially worth 5% of that will be worth £8 million per annum. So it's a significant market opportunity. And if you've looked on the side here, you'll see that we have a very good margin in terms of the work that we're doing there, in terms of its profitability. So the real focus, however, though, is getting into the sales for clinical use. So we want this product to be sold on a widespread basis for the benefit of patients, to be used in treating patients. And the research use sales that I was just describing are very important because they can generate economic return to us, they can offset our ongoing development costs in the business, but crucially they also bring more and more and more cancer centres using our system, all of whom are trying to do research to find out how best to use this for the benefit of patients. They're not doing it to please us, they're doing it because they want to actually solve medical problems for their patients. So the two things we need, I mentioned regulatory authorisation already, but the other thing is patient data. So we're working extremely hard with major cancer centres to get high-level, high-quality clinical evidence of the effectiveness of this technology for patients. And our first clinical application is in ovarian cancer. And we heard Satu talking about endometriosis. Ovarian cancer is a similar kind of, well, it's not really similar, but it's in the same part of the body for the woman. And it's a highly dangerous disease. It's unfortunately known as a silent killer because it's very, very often diagnosed late stage. And the outcome is very poor as a result of that. Therefore, there are women who are having operations for an abnormal pelvic mass. The doctors do not know whether they're dealing with a very dangerous ovarian cancer or whether it's a relatively straightforward operation such as a hysterectomy. What we're doing is producing a blood test, and we're hoping that it's going to have very high sensitivity and specificity. Whereas the existing approaches are, quite frankly, either very, very low on sensitivity or very low on specificity, meaning that it either fails to identify women who've got ovarian cancer or the opposite, false positives, and tells people that they've got ovarian cancer when they haven't. So Medical University of Vienna have used our system and they have found that it's performing extremely well. So we're in the process right now of working with them and some other European cancer centres to put together a study trial to prove this out. So that'll involve several hundreds of patients who are being subjected to operations for abnormal pelvic mass, and we will seek to predict whether they've got ovarian cancer or not ahead of their operations. And that will open up for us a £300 million clinical sales opportunity in ovarian cancer. And we anticipate and we're working towards having the European end of that study finished by the end of next year. So there's a lot to play for in 2016. We've got various other cancer types as well. So nearby we've got the Barts Cancer Institute, and they specialise with our system in prostate cancer. And they have found that in 52 cancer patients, we got 100% of them, we got circulating tumour cells out. So we got the cancer cells in a liquid biopsy successfully in every single patient. That included 8 early stage patients. This is completely changing the paradigm. Historically, you would have expected at best to get 40% of the metastatic prostate cancer patients and probably none or 1 or 2 of the early stage. So that really opens up possibilities, and BARTs are working flat out to actually deliver some therapeutic decision making from those cells. This one here is another one. I'm speeding up so we can have time for questions, but breast cancer is being worked on by University of Southern California, and they have done some phenomenally interesting work on metastatic biopsy. So I mentioned that I'd come back to breast cancer. What they've found in doing a metastatic biopsy, it's extremely difficult. That means, for example, the woman might have a secondary in her liver or in her bones or for a variety of other places, and getting in and cutting out a biopsy in those circumstances is very, very hard. So what USC have done, and they've only published so far the first 4 patients, but what they've done is they've done a whole genome analysis with next generation sequencing of a metastatic biopsy from the operation, so the surgical excision, and they got all the genetic information on that patient's cancer, and then they compared it with a blood test using our system, and hey presto, we got all the same genetic information, which is a real game changer, because that means they're looking at a blood test to take the place of very, very nasty operations. This one is the most recent study. Cancer Research UK Manchester is our longest running key opinion leader. They've been working with our system for 2.5 years. They've got 5 of them installed up in Manchester, and they're the research arm of the Christie Hospital, Europe's largest hospital by number of cancer patients. And they have concluded that our system is ideal, has a unique combination of features, making it suitable for routine clinical analysis. And in the process of doing that, they confirmed that yet again in lung cancer, we've got 100% of patients with circulating tumour cells, and we've greatly outperformed the existing systems. So I think that's a summary for you. I'll come to the last slide. So and Ian, do you want to come and join me? We're at a very, very exciting point now. Starting to make research use sales is great. We're building momentum in that. We're getting closer and closer to clinical application in a very, very large growing market. And to do that, obviously, we're working with some of the leading cancer research centres in the world. And every time we sell a system, there's another one working with it. So we're very excited, and we've got 4 cancers with very good results already, and more to come. Thank you very much indeed.
Questions, anybody? Gentleman at the back, and then one down here at the front after.
Just looking forward to 2016, 2017. Have you got your revenue targets that you can share with us?
Yes, there's a research report up by Edison on our website. In fact, you can find some forecasts they put out. In terms of revenues, it's about 340k to the 30th of April 2016, and the year after is about 2.2 million, made up of principally these sales to the research, or the research use sales that Andrew's been explaining, but in the later year beginning to make sales into the clinical market based on the back of results from the ovarian cancer study as the first application.
I mean, I would say that the immediate sales are looking promising, but the overall target is the big market, and that's what we've got to move towards that. And so the big emphasis is on getting the right clinical information, getting the right support from the major cancer centres, because it's the big game that we're actually going for.
Gentleman down here.
Okay. Probably a question for Ian. Have we got enough funds now to get through to break even, or are we going to have to do another fundraising before we get there?
Okay, so we did a fundraise February, March, and that was very well supported by institutions and shareholders, raised about 8.2 million net. That left us at the year end with about 8.4, just over. We've got other sources of income. There's the final balance, the geometric retention, the sales that we've got, and R&D tax credits as well. Based on a very tightly focused plan that we're following, that's enough to take us through to cash flow break even. What it's not enough for is if we get other applications identified, it doesn't fund the clinical studies that are required to do those other applications. So we're fine, following a very tightly focused plan that's getting the research use sales and developing the clinical application for ovarian cancer and keeping the key opinion leaders working.
At the AGM last summer, you were talking about, sort of off the record it may have been, that you might actually get some revenue from not only selling the equipment and the cassettes, but also other consumables like buffer solutions for washing out things. Do your projected sales figures you put up there include that additional revenue?
No, we've excluded that for the time being. I mean,
I should say they're not our projected sales figures.
Yeah.
He was talking about Edison's, but we don't understand that they've included that.
No. So they'd be upside. The idea is that if your customer's making an order, you want to be able to basically provide them with a starter pack. So you start adding the reagents and so forth and make it easy for them. But a lot of these are world-leading research centres, so they also have access to their own suppliers and preferential rates. So the idea is if somebody wants it, we'll sell it, but it's a modest amount of revenue. It's not the core focus.
Yeah, it's primarily relevant when you get to the clinical market where you tend to provide a reagent kit of everything in one container. So in research use, the research labs have got all these things themselves already. Yeah. So we are selling the instrument and then we're selling the cassettes. And so you get recurring revenue. It's a razor blade model.
Can I just clarify something there? We skipped over a bit about that income stream. Now, Edison's generally get their numbers from you guys, so are you happy with their numbers?
Yeah, they're fine.
Yeah, they replicate your budgeting in a sense.
Well, you always get a few differences because they're their own numbers,
yeah.
But we make sure they're not wildly out in terms of what we think is achievable and what we're going to do.
Okay, thank you.
Could you tell me how much it's holding you back by not having FDA approval, and is it going to make a big boost to your kind of efforts in the States when you get it? Is it creating problems?
We started in March 2014, so we've had a long run at it. It is a significant challenge to get the FDA authorization. I think the crux of your question is to what extent is it impacting our business at the moment? It will certainly impact us once we've got clinical. So I said there were two things: regulatory authorization and clinical evidence. We're in process of putting together the clinical study for Ovarian. So technically speaking, until such time as we have clinical evidence to match the regulatory authorization, it's not holding us back. That said, we would like to get it because it would be a very clear differentiation of our product in the market and would probably hasten the adoption in multiple pharmaceutical trials. So it is a very important and useful thing that we're putting a great deal of effort into getting.
I think the other question is are you going to have to partner to sell this? I mean, is it down the line, are you going to have it going through one of the big companies?
Yeah, so the beauty of this is there's multiple different applications in multiple different cancers, so we can do different routes for different applications. For the research use, at the present moment, we're simply selling directly ourselves, and that's fine because it's got quite a high profile amongst the leading research groups. They know about it and they tend to approach us, so we're beginning to get quite a bit of traction on that. In terms of the clinical applications, the actual analysis of the cells will be done by another system, so it does make sense to actually partner up with such analytical companies, particularly if they've got strong market position already and an installed base. So that is part of our strategy. But we probably will have a mix and match strategy for different parts of the different segments of the market.
Andrew, at the AGM, you said that in your negotiations with the FDA that they kept coming back with new questions. Are those questions beginning to dry up now?
We've had some additional meetings since the AGM, and I'm very pleased with the way it's going. In that we've tried to the FDA has the right, and you could even say the duty, to continue to ask questions until they're satisfied. But in the conversations that we've had, we have attempted to prove that we've answered most of their questions, identify the remaining questions, and specify how we're going to address them. And that has been so far well received. And I have to say the FDA it may not sound like it to third parties, but the FDA are very supportive of what we're doing. So it's not like that. I don't feel as if they're trying to trip us up, but they are putting very significant hurdles. So we've got to prove how well the system works, and we're progressively doing that.
I I think when I pressed you at the AGM, you said they were probably about 2/3 of the way through. Would you be able to estimate how far through they are now?
I think it's a difficult one to say. I don't really know is the answer. There's no point me making a timescale where I'm not really sure.
Anybody got any other
I
questions?
can tell you we're putting every effort we can in to accelerate it, but I can't tell you how quickly it's going to be done.
Most of the drugs so far known to treat cancer or with severe side effects, they're really toxic with loss of hair and the patient losing weight. How, from side effects point of view, how safe these drugs are? How safe these drugs are?
Oh, how safe are the drugs?
Yeah.
Well, the safety and toxicity of the drugs is outside of the scope of what we're doing. What we're trying to do, so a lot of these drugs work on a particular pathway in the cancer, and if the patient's cancer has that pathway, then they might be successful. But if they don't have that pathway, then they're completely useless. So what we're trying to do is to understand the patient's cancer well enough to determine whether that patient would be a responder to the particular drug or not. It's for the regulatory authorities and the drug companies to assess the level of toxicity. That's not something we do.
Thank you.
Time for one more question. Gentleman at the back.
So you've got the ovarian trial going on. Have you got a second clinical application that you're working on to bring, to focus on?
There's multiple clinical applications being worked on, but there's only one that we're heavily investing in and driving ourselves, and that's the ovarian cancer. It's quite possible that when we get good level of evidence and data from key opinion leaders, we may look to actually take on another one. So, and I've shown 4 different cancer types, so there's 3 others that are looking very promising. But at this point in time, we're focused and funded on the ovarian cancer and the research use sales and the FDA.
Thank you, Andrew. Thank you, Ian, as well.
Thank you.